Hello, and welcome to Chapter 7. You have worked all the way through an entire unit in Honors Bio. Congratulations. We started with Chapters 1 and 2. We're skipping Chapters 3, 4, 5, and 6. Don't worry, we'll come back. As we get closer to summer break, those will be the last 4 chapters we do, typically because those are the easiest 4 chapters in the book. And so I think it's safe to say you want all your easy material right at the end of the class, kind of as a celebratory ending of a pretty difficult class. Okay, so we're into Chapter 7, and in this unit, it's all about cytology, meaning it's all about cells and cell biology. We're going to carry that structure dictates function theme throughout every unit in this class, but especially in this chapter. Okay, cell structure and function. There's going to be probably 7 or 8 lectures in this series, in this chapter series. We're going to talk about the surface area to volume ratio in this video. We're going to talk about cell theory in this video. We're going to talk a little bit about microscopes in this video, and basically lay a good, strong foundation for Chapter 7 moving forward. We're going to get in later videos into the difference between prokaryotes and eukaryotes. We're also going to get into the endomembrane organelles. We're going to get into the energy organelles. We're going to get into energy and transport. We're going to get into the cell membrane. We're going to talk a little bit about osmosis diffusion, active transport, and those things. So stay tuned, hold on, let's get started. I'd like to just kind of start by lightening the mood and saying that you are all very much appreciated. So whenever you feel sad, just remember there are billions of cells in your body and all they care about is you. Okay, that's what I typically think about. It gives you a nice, warm, fuzzy feeling inside. So, microscopes. I think everybody has had a chance to look at microscopes or look through a microscope at some point, but if you haven't, you will in this class. I want to talk a little bit about the anatomy of a microscope. We're going to use light microscopes in this class, and light microscopes are all common in that they use light as an energy source. And so you'll see in the microscopes that we use in class or in this diagram that the light is going to travel from the bottom. There's a light bulb down in here. Usually it's protected by some kind of film over the top or some kind of lens. But the light's going to pass through a condenser lens, and so this diagram on the left side of the microscope shows the orientation of the lenses and how the light is bent. Okay, so the light source is going to bend through this condenser lens, which is kind of down here in this housing. It's going to focus on a small point right at the specimen, and this box right here is the stage. And it's called the stage because it's the platform on which the specimen is sat, or the specimen is placed. And so usually there are some clips right in here to hold the specimen or hold the slide securely so that it doesn't kind of rattle around or move around. But it's going to sit on this slide. The specimen's going to be on the slide. It's going to sit on the stage, and the light's going to pass through the condenser lens and focus right on the specimen, which is what you can see right here. Okay, that's the specimen. Once the light goes and focuses on the specimen, it's going to keep traveling and it's going to be condensed again through an objective lens. And most of the microscopes we have in this school have anywhere from 3 to 4 objective lenses. And those objective lenses are going to range from 4 times all the way up to 100 times. It's typically 4 times, 10 times, 40 times objective, and then some of the more expensive microscopes will have 100 times objective lenses, but those are oil immersion lenses, and most of the time in this class, unless your instructor specifies, do not use the 100 times objective because typically we won't need it for the labs we're doing, but it's very important for you to use oil on an oil immersion lens so that you don't scratch the tip, because the lens actually almost sits right on the specimen. Once the light passes through these objective lenses, it's going to go through it's going to go through this nose piece and is going to be condensed further in one of the ocular lenses. Okay, and these are the lenses that you actually look through. So you're going to look through the eyepiece, but inside this eyepiece housing is an ocular lens, and they are typically 10 times magnification. So if we think about total magnification, light is going to be magnified by the ocular lens 10 times, and it's going to be magnified by one of the objective lenses either 4 times, 10 times, or 40 times. And so let's think about total magnification. If I'm using a 4 times objective and I'm looking through a 10 times ocular lens, that's going to be a total magnification of 40 times. Good. Does that make sense? So we have, if we think about a 4 times ocular lens, which is sitting here, and we multiply that by 4 times objective lens, which is sitting right here, okay, 4 times, and we times that by the 10 times ocular lens, which is sitting here, you get a total magnification of 40 times. Now, that means if we're looking at something that's 1 millimeter long, a specimen that's 1 millimeter long, it's actually going to look like or appear to be 40 millimeters long by the time you're looking through the objective. Okay, so we talked about microscopes being or using light as an energy source, especially light microscopes. Okay, we've talked about the light being bent by the lenses. Okay, we've talked about the magnification and the total magnification down here. And one more thing we need to talk about is the resolving power of a microscope. Now, a resolving power is the ability for a microscope to distinguish between two closely spaced points. So if I put these 2 points down here in your lower left-hand screen, you can obviously tell that those two are separate dots, and they're separated by space. Okay, so if I put 2 smaller dots and they're closer together, then it looks more like one individual entity than these. If I do 2 bigger dots and they're a lot more spaced, then it's easier to see that there are 2 separate things. Okay, and so resolving power is the ability for a microscope to distinguish between 2 things that are really closely related, okay, or really closely together. The resolving power that we just talked about within a light microscope is typically maximum 0.2 microns, meaning 0.2 micrometers. Okay, that's about 50, it ranges anywhere from, well, I'll wait until I show you the picture on one of the next slides. Okay, I got a diagram that will show you kind of what the unaided eye looks like. I've got a picture that looks like, we'll show you what a light microscope will resolve to, and then an electron microscope will resolve to. One thing that's really important is that you understand that Robert Hooke invented the microscope in 1665. The year isn't as important as the name. Okay, Robert Hooke is coined or is thought of as inventing the first microscope. Now, it wasn't a microscope like you see them today. It was just really a handheld lens, much like a magnifying glass, okay, but that's considered a microscope because it allowed us to see things that the unaided eye can't, or it allowed us to differentiate between two smaller objects without, by looking at things that are smaller than what we can typically see with the naked eye. He then, once he saw these smaller things with the first microscope, coined the term cell, okay, which is really important because that's what cytology is built on. He coined the term cell because the cork that he was looking at resembled the room in which he slept. Okay, he was a monk, and if you know anything about monasteries or you know anything about jail cells, they are just repeating boxes that aren't very big, that are very simple in their construction, and they're just stacked in a prison or a monastery, one after another, and he slept in one. Now, the one he slept in didn't have bars and didn't have lockdown and didn't have, people in it that were not good to be around. But he thought the cork that he looked at resembled this, and he used the term cell, that these rooms were called because that's what they look like. Okay, and so he called cell, and that's really important. Okay, here is the diagram I told you about. This is the unaided eye, so these are things within this range that we can see without magnification. See, we can obviously human height. I mean, you see other humans walking around, we know their height. That's in the meter range. Okay, it's 1 to 2 meters, which would be about 3 to 6 feet. Length of some nerve cells can be seen. They're anywhere from 0.1 to 1 meter long. So that's a really, really long cell, but we can see those with the naked eye. Chicken eggs, you see, you probably eat those in the mornings. Those are things that you can see with the naked eye. Frog egg is getting towards the smaller end of what we can see with the naked eye, and it's closer to a few millimeters in length. Okay, so when we get into a light microscope, you start seeing most plant and animal cells, and we're going to see using microscopes in the lab that you can see cells that we can't see with the naked eye. You can see them with a light microscope, and you can see them easily. Okay, we can even see some organelles, like mitochondria. We're getting down in the 1 to 2 micron or micrometer length, and so we can see those. Now, electron microscopes are those microscopes that use energy in the form of an electron. So these electron microscopes use energy or use an electron as the energy source. Light microscope use light as an energy source, and these electron microscopes can see things that are much, much, much smaller. And because they're using electrons, which you remember as the smallest subatomic particle, they can see things as small as atoms, which are 0.1 nanometers long, okay, or big. Those are tiny. We can't even begin to fathom to see those with even a light microscope, not to mention an unaided eye. So electron microscopes definitely have their place when we're looking at specific macromolecules like lipids, proteins, carbohydrates, water molecules. Light microscopes have their place when we're looking at specific cellular structures, and an unaided eye obviously has its place because it allows you to get around on a day-to-day basis. Okay, so what is the cell theory? We talked about Robert Hooke gathering all the data known as, gathering the data that called a cell, or eventually calling a cell a cell. But what are the things that came along that contributed to the cell theory? And he wasn't the only one that contributed to these parts, but you absolutely need to know these. I'm going to star this one, and the star refers to all 3 components. And if you notice, these are bold. They've been the only things bolded on this entire PowerPoint. Really, really important for you to know. All living things are composed of cells. Cells are the basic units of structure and function of living things. Basically could be stated as cells are the basic unit of life. And new cells are produced from existing cells. I'm going to show you a picture that has 5 people, and all 5 contributed in their own specific ways to elicit these items as the cell theory. And they have become theory, which means they're more than just a hypothesis. They are basically proven everywhere on Earth. They just can't become cell law because we can't prove it everywhere in the universe. We don't even know if there's other life forms in the universe. So, all living things are composed of cells. That basically means that everything that's living is made of cells. All living things are composed of cells, and they're composed of one or more. We know that there are living unicellular organisms, but all living things are composed of cells. You are composed of cells. You are living. You are watching this video, so I know you're living, and so you're made of cells. Cells are the basic units of life, meaning cells are the smallest thing that is living by itself. We can't say, remember, that atoms are the smallest unit of life, because atoms make up cells, but atoms aren't living, and so cells are the basic unit of life because they are the smallest free-living organisms as well. New cells are produced from existing cells. That means that new cells produce or are produced from old cells. Okay, new cells are produced from old cells. That means spontaneous generation, again, is incorrect, right? Because we can't just put a broth or a stew out or meat and have them breed maggots, right? It's the flies that are landing on the meat that are laying the eggs and are eventually producing the maggots. But the meat itself is not producing the maggots. Old cells have to produce new cells. So, all theory, or the cell theory states, this is kind of one statement that sums up all of them. All living things are cells, and all cells come from pre-existing cells. Okay. Now, on a test or a quiz, when I say state the cell theory, or any of your instructors say state the cell theory, I don't want you just to say this one statement. I want you to actually list 1, 2, and 3. Cells are the basic unit of life. all living things are composed of cells, and all cells come from old cells. Okay, those are the three statements that I want you to know. Sorry about that. These are the 5 people that I want you to know, and this is what we're going to kind of end on in terms of this lecture. So Hooke, remember, was the first one that saw a cell and coined that term. He was the one that invented the microscope, and so he was instrumental in setting these next 4 guys up. Leeuwenhoek was the first one to observe living cells. Remember, Hooke saw or observed cork, which are dead cell walls, and so he just saw basically the boundaries of where the cells were. Leeuwenhoek was the one that actually observed real-life living cells and actually saw them moving, and so he called them animalcules because he thought they were tiny versions of animals. And so Schleiden was the one that basically said all plants are made of cells. Schwann, on the other hand, was the one that said all animals are made of cells, and Virchow is the one that is coined with observing mitosis happening, which is cell division, and he basically contributed that all living things come from cells and that cells make new cells. Okay, so he was the one that saw cell division happening. Cells make new cells. Schleiden, plant cells. Schwann, animal cells. Those two together produced the part of the cell theory that says all living things are made of cells, because Schleiden and plants and Schwann and animals had the entire gamut of organisms. We had both the plant side and the animal side. 2 ways really quickly to remember that. Schleiden, you slide down a slide into wood chips. I mean, if you think about a slide in a park, it's either going to be on grass or wood chips, both of which are composed of cellulose, which is plant-based material. And so if you remember sliding down a slide into plants, you remember that it is plant cells that he worked with. And Schwann, everybody knows what a Schwann is. A Schwann is a majestic goose. It's basically one of those really intimate looking goose. You always see them on a romantic movie setting where the 2 people are walking through a park, and you come to a pond, and there's always a goose that is a Schwann. So we're going to remember Schwann as all animals are made of cells because a Schwann is a majestic goose. And Virchow, again, is the one that worked with cell division, and so he said all living things or all new cells come from old cells. So I think that's where we're going to end. The next lecture will be on cell size and surface area, or surface area to volume ratio. Until next time, see ya.